Okay, so now we're going to talk about lipids. This is topic 6. So lipids are not repeating monomers. Remember, they are not able to make polymers. They are rather macromolecules instead. And they are hydrophobic, obviously, very obviously. Lipoproteins are just a type of protein. They transport cholesterol. They transport cholesterol the plasma membrane. Okay, so that's what I was looking for. Lipoproteins are the ones that will transport cholesterol cargo to plasma membrane and increase or decrease cholesterol concentration. So we'll talk about that in a minute. There's 3 major types of proteins. There are neutral fats, phospholipids, and steroids. So let's talk about neutral fat. The main functions for the neutral fat is the energy storage, cold insulation, and protein inner organs. So energy, isolation for the body, and protection for inner organ. Basically, fatty stuff will protect it. There's 2 types of neutral fats. No, not 2 types. They're made out of 2 kinds of molecules. So neutral fat is made out of 2 kinds of molecules called glycerol and fatty acids. So glycerol is this one right here, this bigger one. And fatty acid is this carbon chain with a carboxyl group. So there is a OH alcohol group on the glycerol and the carboxyl group on the fatty acid chain. And this OH can bond with the carboxyl group to form ester bonds. They will form ester bonds. So ester bond is between the carboxyl group of the acid and the alcohol, like I said, and they can be condensed to produce H2O or hydrolyzed to form. No, it's dehydrolyzed to form and hydrolyzed to break these bonds. It's a fatty acid and glycerol combination. And they can be either a monoglycerol, diglycerol, or triglyceride, depending on how many fatty acid chains. So there's only always one glycerol. This guy here that looks like an alien ship. And the fatty acid chain can be either 1, 2, or 3. And for the membrane, it's usually 2, so it's going to be diglyceride. Or we should call it phosphodiester. Yeah, I think diglyceride and phosphodiester is the same thing. No? What? Okay, somehow they are not diglycerides. Main body fat, long-term storage. So when you see adipose, it means fat. That's what the teacher told me to remember. And let's talk about saturated fats. So there is saturated and unsaturated fats, where saturated fats can hold the maximum, means that they hold the maximum amount of hydrogens. And there are single connected to each other with single covalent bonds, and they're able to bind to their full complement of hydrogen, like I said. And because they form linear molecules, they can easily stack. Yeah, they are linear, so they can easily stack, and that's why they will become solid at room temperature. So it is by van der Waals stabilized bonds, stabilized molecules, and solid at room temperature, like I said. So they are linear. Unsaturated fats are unsaturated fatty acids are liquid at room temperature, and that's because of primarily this double bond between these amino acids. Yeah. So they don't have the full complement of hydrogen atoms. They're unsaturated, so not all the carbons are binded with the number of hydrogen, the maximum number of hydrogen they can bond with, and there's a kink preventing them from stacking well, so that's why they are room temperature, they don't stack well. There's a cis and the trans fat. They can't rotate, they are bent, there's more space required, they're more bulky, so they're liquid. So the cis is to the same side, the hydrogens are pointing on the same sides for the cis. They're easier to digest because they're more kink, so they're more liquid, and these will support membrane flexibility, whereas trans are tighter and they're less kinks and they're linear, so the hydrogen bonds are on opposite sides, and it's usually harmful for the body. EFA, essential fatty acids, are linoleic acids, so omega-6 and omega-3, alpha-linoleic. Yes. So these are essential fatty acids, meaning a human body cannot synthesize, cannot produce, and we must eat them to actually get them. So that's neutral fats, and they're ester bonded. Another fat that's ester bonded are phospholipids, and they are amphiphatic, meaning there is hydrophobic and hydrophilic parts. So hydrophilic is the polar head, and hydrophobic is the non-polar tail. And it is important because the cell membrane is composed of hydrophobic and hydrophilic region. That's because of the phospholipids. Phospholipids' main function is to compose the cell membrane. So the cell membrane is made by phospholipids, and they're also helpful. They also form lipoproteins. Lipoproteins, which I think I mentioned over here. So they transport cholesterol to the plasma membrane. Yeah, those are called lipoproteins. They will form lipoproteins. The structures are 2 fatty acid molecule, 1 glycerol, and 1 phosphate molecule. There isn't that much need of detail to know here, but yeah, 2 fatty acid, 1 glycerol head, and there's also a phosphate group, phosphate molecule in there. That's why there's a charge up there, and the amphipathic. There's a H missing. Amphipathic. Amphipathic. Okay. Oh my goodness. They're what make the double membrane missils, meaning the double membrane. So it will look like this, right? All the membranes, and that's what the soap will break down. So the soap will break down the hydrophilic side, and leaving just the hydrophobic, which will be broken down. I don't think we need to understand how soap will break them down. Yeah. Anyway, they are double membrane. Now let's talk about. Oh wait, we don't talk about that. Okay, let's talk about this actually. Okay, let's come here. Phospholipids, like I said, they can move. So they can move on their axis. They can move laterally. Remember the word laterally. It's not flipping. It's not other definitions. Just whenever you see laterally, that's the right one. In any direction over the surface of the membrane. And they're rare. In rare cases, they can flip. Otherwise, on their axes, 2D movements, they are rotating. Do we not talk about these? Okay, it looks like we don't talk about them yet. I'll come back to it. So let's move on to steroids. So steroids are not by ester bonds. They're by 4-ring sterol groups. Steroids are by 4-ring sterol groups. And this is what the sterol group look like. There's 4 rings of carbons. And they are, that's what we call a sterol group. And there is cholesterol is a type of steroid. And they're a component of plasma cell membrane and modulating the fluidity. So cholesterol, component of the plasma cell. So what it does, cholesterol is really important. It's a really important key for cell membrane is to put together, will squish together all the fatty acids tell if they're moving too much in the heat, so high temperature, or prevent them from packing too tight. So in low temperature, they'll prevent them from packing too tight, and in that case, it's for them to not freeze. So they will make their legs kinked like this if it's too cold, and they will make them straighter if it's too hot, so that they don't melt. So it's all to control the fluidity, the consistency of the membranes. They will help shutter receptor proteins to messenger. Yeah, they also help with messenger proteins. Yeah, other functions that have to do with the cell membrane. Yeah, there's also some bile salts or cholesterols and cholesterol, but I don't know if you need to know them. Some sex hormones are estrogen and testosterone. There's steroids are corticosteroids and calcitriols. Regulates calcium, muscle, neuron, metabolism, carbo what? Anyways, they're called steroid groups. Bile salts absorb and digest other fats. Steroid derivatives. Not sure what that is. So the structures, yeah, so that's the structure of cholesterol, I think. It's good if it's high density, it's bad if it's low density. So low density lipids, low density lipolipids, they bind up in cell wall and make it hard for the blood to flow, so heart attack. So bad if it's low density. If it's high density lipoprotein, cholesterols will pick up the excess cholesterol to the liver. So they will clean up the bloodstream and they will not stuck, and those are good lipoproteins. Lipoproteins for membranes. So lipoprotein will bring the cholesterols to the membranes. Okay, now we're on to cell membrane. So cell is the word found by Robert Hooke. There's not much we need to know about that. So cell, there's the blueprint, there's the boundary, there's the ability to harness energy, there's self-replicating entity independent of life. There's like 10 times that we see this slide already. The function of a cell membrane is to separate the inside from outside. Those are the cell plasma membrane. Component within the cell, only for eukaryotes, so prokaryotes don't have cell membrane. What? What? Oh, okay. So compartment for the cell. So compartment mean the membranes for cells inside a cell. That's only for eukaryotes. But yeah, of course, prokaryotes also have cell membrane. That's for sure. Maintain inside and outside entrance, protection of exchange. Cell membranes are composed of lipid, proteins, and carbohydrates such as glycolipids and glycoproteins. So that's all there is. Lipids include like sterols and cholesterols as well. Proteins, receptors, and other types of proteins. So cell membranes are formed by phospholipids. They're able to move. They're amphipathic. There's motion going on, and like I said, there's summer and winter different comportement, making them less kinked or more kinked for not melting. Increased saturation will decrease fluidity. Increase unsaturated fatty acid will increase fluidity. So it's all about if it's kinked or not. So like I said, cholesterol is able to do their squishing or the spreading. Cholesterols are also another one that's amphipathic. So amphipathic. Oh my goodness. Amphipathic. So cholesterol and phospholipids so far. They are hydrophilic and hydrophilic regions on them, and those cholesterols can bind on the hydrophilic head and hydrophobic tail. Like these. To push them apart or let them get further apart. So the cell membrane is called a mosaic due to its very varied. Oh yeah, so the cholesterol and the phospholipids, those two amphiphalic stuff, are what makes the membrane fluid, by the way. Fluid mosaic cell due to presence of protein. Where is that? Oh well, anyways, we talk about the mosaic because yeah, here's a mosaic. So there is the proteins, the glycoproteins and glycolipids and other proteins that are sticking out. Lots of things are sticking out. Sticking out is the sugar, by the way, of glycolipids and glycolipids and glycoproteins. And it's a mosaic because there's a lot going on in the cell and it's not like homogeneous. Membrane proteins. Yeah, so membrane proteins, there's integral membrane proteins that are integrated into the cell. So they are some hydrophilic and some hydrophobic, whereas there are ones that are peripheral, integral and peripheral membranes. Integral membranes within the membrane. Peripheral is under the membrane. So integral ones are able to do the transport, enzyme signaling, cell recognition, and joining, intracellular, grabbing or moving. Majority is hydrophobic, and the other majority is hydrophilic, cell recognition, grabbing, anchor, specific function, hydrophobic, hydrophilic have different ones. And peripherals are only the ones under the membrane, no phobic regions. So they're all hydrophilic. So they cannot get into the the membrane. So they have to be hydrophilic. So it's all at the head. There are no hydrophobic regions. They're at the hydrophilic regions of the cells. Therefore, cell signaling and some enzymes at the interior of the cells. Yeah, so just remember that these guys are covalently bonded. Covalently bonded. So it's important that cells recognize each other so that they don't kill each other. Continue as a result of a memory of outside of a covalently. Okay, and we're done. We're done. Yay!